I would like to begin by saying thank you to all of you who are here today. We have a lot to cover, but we are really excited about all the issues. The first matter that I'd like for you to be aware of is the fact that this is our inaugural meeting, and we are looking forward to having 4 annual events which build on the intellectual dynamism that we feel will be coming out of this meeting. And one of the things that has really touched me very deeply, but it may not make a whole lot of sense to others unless it's really explained, is the fact that at first glance, it appears that there are many paradoxes in the medical literature, and paradoxes particularly as they relate to underrepresented populations or non-European populations. But what I grabbed hold of, and what I think you will find very interesting, really elucidating and helpful, is the fact that sometimes when we're looking at something that appears to be a paradox, it's not a paradox, it's just that we don't understand it. And it is giving us clues, but we might have to shift our paradigm in order to understand it. So to some degree, this meeting is really about clues, about medical clues that can be taken from a more precise understanding of historical movements, historical events. Our roster of speakers will include geneticists, nutritionists, physicians, historians like myself, medical anthropologists, data specialists, community health workers, and STEM researchers. But when you look at this list, I don't want for you to have a notion that, oh, this is just going to be really broad-based, because in truth, this particular meeting is very focused on one particular goal. And that goal is to find ways of thinking about, find new ways of thinking about how to close America's racial and ethnic health disparities gap. These objectives will become clearer as you listen to today's keynote address given by Texas A&M Vice Chancellor, Dr. Patrick Stover. Also, tomorrow's Research and Action think tanks, led by biologist Denise Perry-Simmons, will provide the opportunity to continue these conversations on an ongoing basis. On slides, I have also designated the particular think tank number that will focus on the actual concept that I am talking about. So let's get started. The focus is 6 overlooked hurdles to achieving diversity in genomic research. But I'd like to begin by clarifying some terms that we're using very commonly. For instance, social determinants of health disparities. What I began to realize was that the issue of racial and ethnic health disparities has many dimensions, but to a large degree, the only dimension that was actually on the table related to social determinants of health disparities. And of course, this is about institutionalized racism. It's about lack of access to healthcare. It's about poverty. It's about the requirements of working class people to put themselves in vulnerable situations and so forth. Now, the point is that these social determinants are profound and they're extraordinarily important. But the point I want to make is that those are not the only reasons why we have this gap, and in fact, what is a growing gap in terms of the benefits of precision medicine, genomic medicine being available to minorities. So in my discussions, I talk about genomic determinants of health disparities. And I will talk in much more detail, but this relates to genetic adaptations to unique ecological environments of particular ecological niche populations. And I will give more detail about that later. Okay, now I have on this list social determinants, racial determinants of health disparities. But if you can see it, I put a little red line through it because one of the things that I want for you to truly understand as you move away from this conference is that there's no such thing as racial determinants of health disparities. Now, let me back up a bit because I think we have to differentiate medical science and the precision that is absolutely required from our political environment, from the political needs, social needs, to identify issues of discrimination and other factors that need to be remediated. And the only way they will be remediated is if there is a way of targeting those who've been victimized. So the point I'm making is that race is not scientific. It should not be part of the genomic picture, but in social and political terms, it's real and it has value. One last point I would like to make in this regard, and that is about the nature of the discussion at this particular point. I'm hoping that we can change the nature of the discussion. And what I mean by that is in several medical journals, I've recently been reading various discussions about colorblindness on one side versus racialized medicine on the other side. And the point of these discussions is essentially to take aside, I don't believe people should be differentiated by race, or, I think maybe it's a good idea to differentiate people by race. Okay, well, the point I want to make here is that American medicine is already racialized medicine because in ways that we have not been conscious of, it is contoured to the very precise genotype of only one genetic population, which is Europeans. So the point I'm making here is that even the side who argues for colorblindness, they have to be extremely careful because arguing to impose the genetic values of a single population on the entire world is not colorblindness. It's just a lack of truly understanding the challenges that we're dealing with. What I want to focus on at this particular moment is one of the dynamics of that medieval period, the trans-Saharan gold salt trade. Now, the reason I want to focus on it is because we begin to see extraordinary health implications that have really been overlooked until this point. So what am I getting at? Well, firstly, the fact is that you had a large population that was willing to trade the gold that they were mining for salt. Now, why would they do that? They did that because they just so happened to be living in an extremely hot tropical environment that was also one of the most sodium deficient on the planet. And we're talking about the interior of West Africa, not the coast. Coastal populations have have no problems really with salt. But what is the significance of this? It so happens that the peasant population in these sodium deficient areas, they had no connection to this gold salt trade. Most peasant farmers in the interior of West Africa had never even tasted table salt. They flavored their food with the burnt ashes of millet and other vegetables and so forth. And as a consequence, when we look at this group, it turns out that they were functioning on 200 milligrams of sodium per day. But that seems rather extraordinary, especially because other European populations, coastal African populations, they were functioning on about 5,000. But here I'm making a point, and that relates to the fact that, you know, over the course of thousands of years, this population had to adapt to a very unique ecological niche, or otherwise they never would have survived. And so here we begin to see the intersection of history and health, because if you look at the medical literature, 75% of adult African Americans suffer from hypertension, and many of those sufferers suffer from a very specific kind of hypertension, which is salt sensitive. And 75% in, you know, other populations, it might be as high as 25%. But I mean, this is really quite extraordinary. But the problem is that the medical literature has offered all kinds of fascinating theories, and some of them very, very brutal and ugly about the slave trade. But nevertheless, they weren't necessarily getting to a point which is really quite simple for a historian. And that is the fact that if you have a population whose ancestors lived sufficiently healthy lives that they could be kidnapped and taken thousands of miles away and maintain their ability to be highly skilled farmers and so forth, and they're only functioning on 200 milligrams of sodium per day because they are genetically adapted to that environment, then in a sense, what do you expect in the United States, where everybody is consuming on average 3,400 milligrams of sodium per day? So the point I'm making is that so many medical issues are so complex with thousands of variables. And when we're dealing with genomic studies, sometimes we're dealing with billions of variables. But when we look at historical issues with some level of precision, we can, at least in a couple of cases, find answers to questions that really had only 2 or 3 variables. And this is the one of the important points that I want to make. So in this particular instance, coastal Africans were consuming 5,000 milligrams. Interior Africans were consuming 200 milligrams. Most Europeans consumed about 5,000 milligrams. This is before refrigeration, so it was reduced a bit, but people generally eat too much salt. Okay, but in any case, here we see a very important example of the reason race is not scientifically valid, because to someone looking at race, coastal West African from Nigeria or even from Kenya or wherever, they're not going to be differentiated from West Africans from the interior. And yet their sodium metabolism is going to be much more similar to that of Europeans. And the situation of interior West Africans, their sodium metabolism is going to be much closer to several somewhat isolated tribes in the Amazon. But the point here is I make reference to ecological niche populations as a way of recognizing the fact that there are many cases where populations have to adapt to the food environment that they have available to them and the climatic environment that they have available to them. And over the course of thousands of years, it essentially means that they are become much more efficient digestively and metabolically in dealing with what their ancestors had been dealing with for several thousand years. Okay, so here I'm talking about these various and sundry hurdles to achieving diversity. The first one I want to focus on is what I call the one-size-fits-all fallacy, or how I almost ended up on a dialysis machine. In 2008, I was a Fulbright visiting professor in Japan, And it was just a really, really wonderful experience. But in any case, about 2 weeks before I was supposed to go back home to Texas, I decided to take advantage of their extremely wonderful health system. So in doing so, I made an appointment, I went to the doctor, and they did some tests and so forth. And then they called me back. They sounded kind of somber. And when they called me back, this doctor, he had such a kind, gentle voice in his sort of accented English. But essentially he said, you know, lady, you've got renal failure and you need to take care of this immediately because it is your life at stake. And so, of course, I was really miserable. I mean, what a thing to find out. But I get back to Texas, and the first thing I do, I mean, the very first day, I call my primary care physician. I make an appointment with him right away because I tell him it's an extreme emergency. And so I tell him that, oh, it looks like I'm dying of kidney failure. And he starts to laugh. Terror, I mean, really bad, what do you call it? Bedside manner. And then he reaches into a file and pulls out a form, and it's a lab form. And in the upper left-hand corner, it has checkoffs for race and ethnicity and so forth. Well, it turns out he had checked off African American. And this was the reason for the misdiagnosis, because Japan is more of a monoethnic society. So all of the renal values are going to be very similar. But here in the United States, I mean, this is the precious gift of diversity we have within our society, but it also means that in certain cases, we have to understand ancestral differences. And so the reality is that I really began to understand the importance of stratifying in instances where there really are ancestral differences. And in most instances there aren't, but if they are, that can be extremely important, because what renal specialists in the United States finally realized was that if they did not stratify these values, most adult African Americans who were healthy would be on dialysis machines. So this was a situation in which they had to learn rather quickly that something had to be done. But you'll see from the rest of my conversation here that this understanding has not really permeated the medical community and the medical research community. Okay, so one of the most counterintuitive discoveries of recent genomics is the fact that disease causative gene variants can be population specific. And I'm very pleased to say that we'll be hearing more on the subject from Dr. Georgia Dunstan, who's founding director of the National Human Genome Center at Howard University. But this is what I'm talking about. I really do not believe that scientists in 2003, when the Human Genome Project was released, that they understood this very sort of counterintuitive piece of human biology. And that was the fact that different genetic populations can have the same symptoms, but the triggering mechanism will be a different gene variant. And I began to appreciate the challenges created by that fact when I noted that there were more and more reports of those African Americans who at least have the privilege to use genomic medicine, which in many cases, many people are too poor to do, their lab reports would come back VUS. VUS means variant of unknown significance. It would either have that designation or the designation would be rare. And this is where I realized, hey, but we have a serious problem, because the reason it's rare or VUS is merely because it's not on the European genome. And it so happens that Africans have significantly more gene variants because they carry the ancestral genome. So one of the issues I noticed, which was kind of fascinating, was that in 2009, the Chinese decided that they really did not want to wait for the West to figure out what to do about its European-centered genome. And they began stratifying their own. And what they did, which I think is interesting and instructive, is that the primary population are the Han Chinese. But when it comes to gene variants that are triggering diseases and so forth, oftentimes it's really a particular ecological niche that has adapted gene variants to the specific use. So what the Chinese finally figured out was the fact that stratifying the Han genome from the European genome was really the absolute beginning, because what they had to do if they really wanted to create benefits for people in their society was to stratify a reference genome for the Northern Han, and a different reference genome for the Southern Han, and a different reference genome for the Beijing Han. And so what all of this adds up to is something that I don't think we were really anticipating at the beginning of the Human Genome Project. And that is homogeneity in a diverse world. And what I mean by this is the fact that the more genotypically homogeneous a reference genome is, the more successful it is in genome-wide association studies. And this is because of the fact that different populations will, in some cases, have different gene variants performing the same function, and therefore will end up with people of European ancestry gaining more and more and more use from our reference genome, but people of non-European ancestry, even when we're trying to recruit more of them, as long as they're just mixed in with the Europeans, then the problem is going to be one where you have variants of unknown significance. Okay, and I think a prime example of this, and what really got me started on it, was research on lactose intolerance and lactase persistence and non-persistence and so forth. Professor Sarah Tishkoff of the University of Pennsylvania did a really, really extraordinary work in 2009, in which she was able to identify the gene variant that was a gain of function gene variant, allowing certain populations to digest the lactose in milk. But this is where her research took a very strange turn. As she did it, she began to realize that Northern Europeans had, those Northern Europeans who were lactase persistent or lactose tolerant, the gene variant that was causing it was 13.9.10. But in Kazakhstanis and Northern Indians, it was 22.0.1.8. And then remember what I said about our ancestral genome. It's significantly larger in terms of gene variants. So when she began looking at the situation in Africa, Sudanese had a gene variant 13.9.07 that was giving certain ones of them, those who were lactose tolerant, it was giving them that particular benefit. However, Kenyans who were lactose tolerant had 13.9.15, and Tanzanians, who are their next door neighbors, had 14.0.10. So the situation was that it was becoming more and more complex. Fortunately, today, Dr. Kim Pruitt from the National Library of Medicine will be focusing on some of these issues related to the reference genome and different approaches that we may be interested in taking. Let me also mention that African Americans of slave descent have a particular genomic profile. And this is because there have been so many of these Ancestry.com, 23andMe sorts of things where people send off for their ancestry and whatever. So over the course of time, statisticians have noticed a kind of average. And that creates a range, but it's a genomic profile. So 75% of on average, 75% of African Americans of slave origin are 75% of their DNA is Niger Kordofanian or Bantu speaking West African. However, 24% is Northern European, 1% is Native American. And I mention this particular point here because many populations are admixed. So now we have the tools to be able to delineate these admixtures. And those tools can be used to help us also find gene variants. Because in the case of African Americans, I'm just going to offer a hypothesis, but I like my hypothesis. And that is the fact that, interestingly, 24% of African Americans are lactase persistent, and all African Americans have 24% Northern European, not all, but in general, Northern European DNA. And so it is in all probability the case that even though you have all these different gene variants in Africa, it's probably the European gene variant that you would find in African Americans. But let's consider for a few moments what we do to fix the new challenges that we're beginning to see so clearly created by the fact that many diseases are turning out to be population specific. The answer may be that we have to take several tracks at the same time. There are efforts that are being made that have been established over the course of the last several years, which add new sequences to the human reference genome. And in fact, there's been a very sophisticated process of adding more and more sequences to this particular genome. So that needs to continue, and it's really of critical importance. But I think there has to be another approach, which is simultaneous, and that is focusing on stratifying populations by ancestry in cases where it becomes clear that the certain genetic variants that are causing certain diseases are in fact population specific. And one of the things I think about as I relate to this is the fact that even scientists sometimes really don't understand what we have learned about human genetic diversity, in the sense that our species as Homo sapiens had a certain amount of genetic diversity. But then around 60,000 years ago, we began to have a series of migrations, actually one primary migration. It's called L3, the L3 branch. Well, this is one branch of the species that has migrated out of Africa. And so that particular branch did not carry or did not retain every single one of the gene variants that are part of our species. But we will find these various gene variants in different African populations. Well, the reason I make this point is because we may need to reconsider whether adding snippets of African DNA to its European subset will offer the needed benefits, because here's where we really begin to see this kind of radical understanding of race that comes out of genomics, but that we're not really absorbing. And I'm very excited to say that we have several specialists, specifically Dr. Jeffrey Long, who has conceptualized these new issues, as well as Dr. Neil Hanchard and Dr. Rick Kittles. But here's this little chart, which I borrowed from Dr. Jeffrey Long. And it's kind of startling for people who think, well, if you're from Scandinavia and you have blonde hair and blue eyes, it's because you have those gene variants. And then you're from the Congo, and so you have dark skin and kinky hair and whatever, and you have those gene variants. But what the latest research has shown us is that that's not even what is happening with the human population. What happens is that the ancestral genome has the full range of genetic variants. So therefore, when you have a population moving from sub-Saharan Africa to Sweden, it is not necessarily, or in fact, it's not the case, that the Swedish people are evolving new mutations and so forth. The Swedish people are, over the course of time, bringing to the surface recessive gene variants that the Africans carry. But the problem is we don't see it in Africans, even though, if we were looking at 1 billion people, you might have a couple with blue eyes, but they wouldn't be the same as the ones with blonde hair and whatever. So we'd never put it together because there's no ecological niche in Africa that demands it for survival. And this notion, of subsets, races as subsets, it's something that we have to give a great deal more thought to. Okay, here I'm talking about hurdles, and one of the issues that I think we need to reflect on very, very seriously relates to precision medicine and precision nutrition. And this is the fact that they only work when they are precise. They only work when they are precise. The United States has spent more than 2 decades and several trillions of dollars to develop ever higher levels of data sophistication and complexity in linking disease to variants in our current reference genome. But here's the problem. As his work becomes more and more precise, it becomes more effective, but more effective for the population represented within the reference genome. So we have a situation where precision medicine is making just enormous breakthroughs and discoveries and so forth. But every time you see one of these discoveries in the media, you really have to ask yourself, for whom? For whom? Because the reality is that the precision in the reference genome as it relates to Europeans is so vitally important that even when we're adding snippets from various other populations, there's no precision there. I mean, there are a whole lot of people on this planet. So this is why we have to take this issue of precision much more carefully. And so I'm just laying out ideas, but these issues I think have to be grappled with in serious ways. Okay, another issue that bothers me in a way is the fact that oftentimes, even within academic communities, we have a tendency to confuse genetic diversity with ethnic diversity, or to conflate both of these. And it doesn't work. Ethnic diversity is putting forth the effort to bring more underrepresented groups into the American mainstream. And this is something that's very precious to my heart, and I hope to many of you all as well. But genetic diversity is counting up the number of gene variants, genetic variants, and single nucleotide polymorphisms in a particular species, counting up the number, the genetic variation. And so it so happens, where's the genetic variation in the human species? It is contained on, carried by people inhabiting the African continent. So the kind of ironic thing is that in terms of ethnic diversity, if you want to have a room full of diverse people, you'll have whites and blacks and Asians and Native Americans and so forth and so on. But if you want to have genetic diversity, then you're going to have a room full of Africans. And we need to understand this, because at this particular point, after all that's been done genomically, it remains the case that only 3% of the African genome has been sequenced, and approximately 88% of the European genome. So here again, we see where there is precision and where there really won't be precision. One of my final points is the exciting efforts that go into looking for medical paradoxes, and I call it a researcher's gold mine. West Africans have exceptionally low rates of osteoporosis, even though they live in the tsetse belt. And the tsetse belt is, these little insects, these tsetse flies. And so it's not possible to breed beasts of burden. There is no dairy farming except in certain outlying marginal areas and so forth. Okay, so I was doing research several years ago, and the data kept coming up saying that West Africans were virtually immune to osteoporosis. But then I thought, I can't publish this because nobody will believe it. And especially because there aren't sufficient medical institutions and resources that can really back up something that sounds really radical. Okay, so I just kind of let it go. But then I woke up one morning and I realized, oh, wait, I have some data. Who are African Americans of slave descent? They are 75% West African DNA. And so when I began to look at the profile of African Americans and osteoporosis, what I found was the fact that of all the demographic groups in the United States, they had, in fact, the lowest rate. And also, African Americans of slave descent have strong bone mineral density and so forth. Okay, so here we have a situation where 75% of African Americans are lactose intolerant, and so they are not drinking milk. And by federal nutritional standards, this particular demographic population is considered to be calcium deficient. Peeling away the layers of this particular medical paradox, I came across something quite extraordinary. And that was the fact that researchers, and some of these researchers you will be talking with tomorrow, had already done a considerable amount of work on a particular gene, human gene, TRPV6. It's a calcium ion channel. And it so happens that the African variant of this calcium ion channel is exceedingly more absorbent of calcium than the European variant. So all of a sudden, things were beginning to make more sense. Oh, if you happen to have TRPV6A, then you do not need as much calcium as if you have TRPV6B. But this was really just the beginning of this journey. I sort of put together my little diagram here, and what it was essentially saying is that when Europeans, people of European ancestry, consume 1,000 milligrams of calcium, they will lose at least 100 in their urine. Whereas if an African-American male is consuming 800, then they will only be losing 35 because they'll be holding on to much more of the calcium. Okay, but here we begin to have the problem. How does this matter medically? And the answer is cancer. The answer is cancer. The uncontrolled proliferation of TRPV6 is a biomarker for several aggressive subtypes of cancer, which include those that African Americans have unusually high mortality rates from and high susceptibilities to. This includes metastatic prostate cancer, and African American males are dying at 4 times the rate of other males. And it includes triple negative breast cancer, where 2 to 3 times more African American females are dying. Well, here is the point I'm making. Calcium is one of the most highly regulated substances in the human body. And there's more and more research that's beginning to suggest that the overconsumption of calcium can in fact trigger this proliferation of protein, of TRPV6 protein. So here we have a situation where these are issues that absolutely have to be looked at much, much more closely. And fortunately, you will be hearing tomorrow from Dr. Alexander Sobolewski and Dr. Patricia Francis Lyon, both of who are doing pioneering research in this particular area. But I must say, it's only now that we are stratifying populations to some degree that we're beginning to even see connections that were not so obvious before. I've had the great fortune to work with a colleague at UNT, Helen Wang. And so I put together this cute little pictorial graphic thing, trying to say that, oh, you stratify populations and you use the admixture ratios in order to determine in areas where nutrient values might be critical, how much each needs, as opposed to simply assuming that everybody needs what Europeans need. However, to my fortune, Dr. Wang listened to me and she listened to me and so forth. And so she put together this really excellent formula that automatically universalizes it, whereas I would be going on with pictures forever. Okay, so here my point is that there are clues upon clues that many of which we are missing. I think Dr. Stover will be talking about some of these nutritional clues. We'll also have Dr. McCall and Dr. Anderson and Dr. Hidalgo talking about clues. But this is what we have to understand in terms of becoming more precise. And my last point is simply that America is not just multi-ethnic or multiracial. America is a multi-genomic nation, and we have to reckon with that. Thank you very much.